Hello, everyone. Welcome to the Tisch Cancer Institute seminar series. My name is Ken O'Neill. I'm today's host. We are incredibly fortunate to have as our speaker today, Elaine Martis from Nationwide Children's Hospital. Before I introduce Dr. Martis, just a couple of quick ground rules. First, please, as Dr. Martis is presenting, remember to mute your microphones. You know, there are many entertaining things that are happening in all of our backgrounds, but but perhaps, you know, sort of like Las Vegas, things are better left there. Second, if you have questions that come up over the course of Dr. Martis's talk, please type them in the chat box at the bottom of your Zoom window. After Dr. Martis is finished with her presentation, I'll just read to her the questions and she'll answer them for us. Okay, so those are the only ground rules. And so now let's get started. Our speaker today, of course, is Elaine Martis from Nationwide Children's Hospital, where she is the co-executive director of the Steve and Cindy Rasmussen Institute for Genomic Medicine, the Steve and Cindy Rasmussen Nationwide Foundation Endowed Chair in Genomic Medicine, and a professor of medicine at The Ohio State University School of Medicine. Dr. Martis is originally from Nebraska, but despite being from Nebraska, she went to Oklahoma for her undergraduate and graduate work. She did her PhD at the University of Oklahoma in chemistry and biochemistry under Bruce Rowe. After she completed her PhD, she was a staff scientist at Bio-Rad for 4 years. And then in 1993, she made the fateful decision to move to Washington University in St. Louis, where she remained until 2016. Eventually becoming first co-director of the McDonald Genome Institute, the Robert E. and Louise F. Dunn Distinguished Professor of Medicine, and a professor in the Department of Genetics and Departments of Genetics and Molecular Microbiology. You know, and of course, Dr. Martis has published over 350 papers. She has won innumerable honors, including she's a fellow of the Academy of the AACR. Last year, she was elected to the National Academy of Science, and currently she's the president of the AACR, recently overseeing in her virtual living room a very successful virtual meeting with over 61,000 participants. Dr. Martis is remarkable. She's one of my true heroes. She's made many groundbreaking discoveries in the fields of cancer genetics, infectious disease, and other complex traits, many of which I feel are paradigm shifting and really change the way that, you know, I and many others in the field think about, you know, just things. In cancer specifically, she has influenced how we think about cancer etiology, the emergence of drug resistance, and sort of targeting cancer therapies to molecular alterations and other alterations found in cancer cells. Today, since moving to Nationwide in 2016, she's really focused her work on pediatric cancers. And in fact, today she'll tell us about what she's been doing at Nationwide on precision medicine for pediatric cancers. So without further ado, Elaine, you're up. And thank you for joining us today virtually.
Okay, great. Thanks so much, Ken, for the nice introduction, and thanks for the invitation to speak today. As Ken mentioned, I'll be talking about this program we rolled out a few years ago in precision medicine in the pediatric cancer space. Where I'd like to start today is really just to contrast where we're at with adult precision cancer medicine, which I think was captured at least to first approximation by this review that Mike Berger and I wrote a couple years ago in Nature Reviews Clinical Oncology. Just sort of describing all of the ways in which a standard next generation sequencing based comparison of tumor to normal has really, you know, developed into this precision diagnostic approach, now including aspects of immunotherapy, checkpoint blockade, and neoantigen prediction for personalized vaccines. So this is a very rich sort of standard of care, if you will, that in large part has not translated so well to the pediatric cancer setting. So why is that? Well, there are a number of reasons. First of all, pediatric cancer is a rare disease, so sort of getting the heft and numbers behind significantly mutated genes and the development of targeted therapies to address those has been, I would argue, much slower. We do know that there are a small subset of genes that are frequently mutated both in pediatric and adult cancers, and so would be amenable to treatment with FDA-approved drugs from the adult setting, but this is sometimes difficult in the pediatric setting, largely due to challenges of dosage, unknown adverse event profiles, and even, unfortunately, a willingness for insurance reimbursement. In addition, the enrollment onto clinical trials has patient age as an exclusion criterion. This is changing now with the Race for Children Act, but I think change will be slow. And we don't really understand in many ways cancer biology in the pediatric setting, especially for these very rare cancer types. More recently, as I mentioned, genomics, including neoantigen prediction, tumor mutational burden, other things, really in terms of the application of checkpoint blockade is really not considered terribly appropriate. Although we and others have seen kids with constitutional mismatch repair defects that have contributed to their likelihood to be treated and respond to checkpoint blockade. But in general, it's a rarity. And essentially every patient is an N of 1. And so I think this raises a number of significant issues that I'll talk about today. On the other hand, pediatric cancer is the leading cause of death by disease in the developed world in children over 1 year of age. And much like in the adult setting, pediatric brain cancers are actually the leading cause of death by cancer in pediatric patients rather. So we have a significant focus in my research laboratory on CNS cancers, which I talked about yesterday for the New York Academy of Sciences immunotherapy symposium that's also going on right now. So we have had large-scale genomic discovery happening in the pediatric cancer setting, somewhat coincident with the adult TCGA, ICGC programs that you all are probably familiar with. What have we learned from this? This is really just a summary report in 2012 from a large-scale project that we did in collaboration with St. Jude back in the day when we were at Washington University. And what this summary statistic really shows is that across leukemias, solid tumors, and brain tumors, you're typically rarely finding point mutations when you look at very extensive landscapes. So this was all done by whole genome sequencing. But in general, the number of mutations is extraordinarily low. And really what's much more common are gene fusion drivers contributing to the onset of the cancer by virtue of translocations or inversions typically, as well as a considerable contribution from epigenetic drivers. So changes in histone proteins, changes in methylation, et cetera, that really in aggregate affect a modified transcriptional program to drive cancer onset and progression. So I'll talk about the transcriptome a lot today because I feel like it's really become a very rich information source for the pediatric precision medicine setting, but still has some challenges as well. Lastly, what we identified in this study is that the germline contribution to pediatric cancer predisposition is actually much more significant than previously thought. Typically, you would have heard a 2% maybe a contribution to predisposition. What we're really identified and others have identified subsequently is more in the neighborhood of 10% or sometimes higher, depending upon whose study you look at in terms of germline predisposition. This is the study that I was referring to earlier in terms of this very small overlap between adult and pediatric cancer. So on the left-hand side, you can see the huge disparity in the mutational burden that I mentioned. On the right-hand side, you can see when you compute significantly mutated genes from both adult cancer studies and pediatrics, that this very small overlap is, illustrated in this gray oval below. And if you really look carefully at many of those gene names, a lot of them are, tumor suppressor genes. So not immediately, if you will, applicable for like a targeted inhibitor, small molecule therapy, et cetera. So this just, I think, exemplifies the challenges that we're up against. So to address these challenges, when we moved to Nationwide Children's Hospital, our real mandate in moving there was to start to introduce genomics into the practice of medicine. And one of the most obvious places to start, of course, is in cancer, just for the reasons that I stated in terms of the contribution to pediatric deaths, as well as the fact that we really understood that in the context of the intimacy of a tumor to normal comparison, you have an incredible amount of power. And we really wanted to begin to explore that in the translational setting. So we introduced an IRB approved protocol, which I'll tell you about today. The graph here, or the figure, is really meant to illustrate the different steps that we go through in this process. And I don't want to spend a lot of time on the nuts and bolts of how this works. I think people are pretty familiar with the need to consent, the need to get tissue and blood, and to study it in ways that I'll describe in great detail. One of the things that I think we did very well initially with this protocol, just to point it out, is at the upper left-hand side of the slide, which is that we made it easy for our oncology providers and surgical providers to nominate patients onto this protocol. We simplified this in REDCap, which captures all the other information that I'll tell you about today. And we really just made it a one-page form with very few fields that needed to be filled out. Most of it is clickable radio buttons. And once this nomination form is completed by the provider or their CRO or whoever is authorized to do that to nominate each child, then the study team is immediately notified electronically by REDCap. We can go in, review that nomination, vote, and as soon as we have 3 study team members that have agreed to the nomination, then our genetic counselor is alerted so that they can approach the family about the study, and off we go. So this made it very facile, as I said, to enroll patients, and you'll see the summary statistics here in a minute that show that this has actually had a pretty significant uptake since we introduced it in late January of 2018. One thing you may be wondering is, okay, so what do you do with these samples once you get them? And what you can see is described here is that we have a number of processes, primarily just this tumor to normal comparison using exome capture. So exomes means every gene in the genome. So an unbiased look, but also including into that capture reagent probes that actually tile along each chromosome. So it's kind of like having high depth coverage on the genes, but then also coverage across the chromosomes so that you can compute copy number, which in pediatrics actually turns out in many cases to be prognostic in terms of specific genes that are amplified, deleted, et cetera. So we really need to have that information, but we really didn't want to pay still the cost that's required to generate whole genome data at high depth. And so this to us was a reasonable compromise because those copy number probes just add about 1.5 megabases to the total genomic space that we're pulling down in this exome comparison of tumor to normal. As you can see from this slide, that currently is not in the CLIA setting, although we do run the molecular genetics and cytogenetics CAP-CLIA lab at our hospital. The plan is to make this an orderable test with CAP-CLIA validation in place by the end of this calendar year. So it will transition, the tumor normal exome will transition into the clinical space. RNA-seq will be a little bit further behind. One of the reasons for that, as I'll talk about today, is there are a myriad of different ways to analyze the RNA-seq data. And the second reason is listed below, which is that we already have CAP-CLIA validated ArcherDX assays for one of the most important things that we can find from RNA, not the only thing, but one of the most important things, which are these fusion drivers. So this is an insurance billable test. We've had it in place for over 6 months now, and it's got a fairly fast turnaround time as well for the clinical sign out. So we can still offer that fusion detection alongside the tumor normal exome by the end of this calendar year. Another thing that we've really pioneered since we came to Nationwide Children's in this protocol, and with the help of Peter White, our Senior Director of Computational Genomics, and his incredible team, is the ability to compute all of our data in the Amazon Web Services cloud. So you can see here on the top panel our enhanced exome sequencing approach. On the bottom panel, our tumor-only RNA sequencing approach, and reading from left to right, sort of digital images of what's happening, where the picture of Winston Churchill is meant to represent our rapid alignment algorithm called Churchill. So these Churchill-based pipelines then combine a number of different analytical tools, especially in the RNA space, for doing things like fusion detection, variant calling, tumor microenvironment deconvolution, pathway analysis, et cetera. And I'll talk about some of these specifically in my next remarks. But this is really a rapid turnaround pipeline because of the power of the cloud-based computing and the fact that we can utilize as much capacity as is available to us to turn around these analyses very, very quickly. And typically, if you're wondering about the turnaround time, this is, we're getting information back to providers in about 2 to 3 weeks on average for this research-based protocol. So once we have all of this information in place, then we consult a variety of different databases. I wanted to just briefly give a shout out to some of these. Of course, the PCAN database and the associated tools offered by St. Jude are really transformational and represent a nice information source to go to when we've identified a somatic alteration in a specific gene, especially if it's something we've not seen before. Next, I wanted to sort of focus a little bit on fusion detection. So I told you that we already run the ArcherDX assays in CAP-CLIA, but we're generating RNA-seq from the tumor. And one of the things that we want to be able to determine is what fusions are there. I think the complexity of this figure ought to tell you at least one takeaway message, which is fusion detection is hard. And in particular, if you look at this figure, the orange boxes that are sort of across the middle and the names that are in them represent the names of different fusion detection algorithms. And so in NGS analysis, anytime something is difficult to detect without with high precision, we typically use this multi-algorithm examination and then a voting mechanism that allows us to combine the results from these multiple fusion callers, as you can see in the blue below, along with some level of prioritization that tells us that if multiple callers actually find a fusion, the likelihood that it's real is very, very high. And I'll talk more about this in a second. But then we basically take these downstream to do some knowledge-based clinical filtering. Has the fusion been seen before, and what kind of cancers, et cetera? And then ultimately, we do have the ability to perform a validation, actually a confirmation of these fusions in our CAP-CLIA lab that can be written into the patient's medical record. This just gives you an idea of some early data that we had comparing the different fusion callers. And the idea behind this was two things. One, to figure out which fusion callers were typically right and which fusion callers were typically not right or didn't return an answer. And so by virtue of this over time, we hope to not have 7 callers running all the time. We'll winnow that down to the 4 or 5 that are the best performers. But in the reality of what we've been able to identify, as you can see from this descriptor, and this is a sort of a preliminary result that's a few months old now. And at the time, we had about 131 cases all the way through analysis, different types of tissue sites, as you can see. And for the 31% of those cases that had a fusion detected, when we took it to the CAP-CLIA lab for confirmation, we were at 100%. So we feel like the approach overall is working, and we just need some refinement over time. So we're not running all of these callers, some of which take more time than others, and some of which, as I just showed you, are sometimes not returning any information. And so those wouldn't be worth the CPUs required to run them. So this is a work in progress. We're actually writing up our fusion analysis, and we're hoping to submit that soon for the total number of cases that I'll show you in a minute. Another database that we've been using and now contributing data into is run at UC Santa Cruz by David Hausler's group, and Olena Morozova-Vasquez in particular is in charge of this treehouse effort, which has been funded by a variety of pediatric cancer foundations. A really, I think, transformational capability here, because essentially what you bring to the treehouse can be as small as a single data set from RNA-seq from a single patient, just the top 500 expressed genes. And using the tools and the database that they've built, you can actually make comparisons of your patient based on RNA sequencing to the multitude of other patients that are available at the treehouse, and performing a variety of different analyses, like grouping that patient with other patients having a known diagnosis, for example, can sometimes help you understand patients with an indeterminate diagnosis by conventional pathology, for example. So there are a variety of mechanisms by which we use the treehouse data, and I'll try to illustrate some of those throughout the rest of the talk. One area that we're also evaluating, which I'll show you some evidence for, that uses the treehouse cohort, as you can see on this leftmost figure, is the gene expression outlier analysis. And what this is really attempt to do is to compare the top expressed genes that are statistically overexpressed relative to other genes in that patient's genome, using the treehouse cohort again as a comparator. And so this sort of multiplex analysis workflow that you see on the left-hand side of the slide ultimately ends up telling us differential expression, as well as the notion that genes that have extraordinarily high expression levels, like this example shown here with FGFR4, may actually render down to a druggability target that could be, for example, confirmed by immunohistochemistry for using an FGFR antibody, for example. That might get that patient onto a therapeutic trial or an already approved drug using compassionate use or insurance, whatever works. And we've had numerous examples of this in our work so far. So the gene expression analysis is important. I think it also goes forward into ingenuity pathway analysis, which I think I have an example to show you as well. But at the end of the day, where we really are finding difficulty with these results moving into the clinical setting is how do we confirm them? And at this point in time, really, the availability of immunohistochemistry is kind of the rate limiting step. And now turning our attention to the tumor microenvironment, just briefly, we want to understand what immune cell types may be present there. This is a tool that I'm using a lot in my research, using the data that have been generated from this protocol, but actually looking at longitudinal samples. So kids that have a primary occurrence of their cancer and then a recurrent cancer, we can often use CyberSort, which again just takes RNA expression information in as the data source, and uses it to compare to this expression matrix that's been pre-generated by the CyberSort folks who are based at Stanford. And essentially comes up with the relative fraction of all 22 different immune cell types that may be present in the tumor mass, as well as a global p-value for these signatures. And this can tell us about changes to the tumor microenvironment that might make it more or less amenable even to immunotherapy approaches. So just to finish this part of the talk then, I just wanna sort of bring you up to where we are currently. You can see that we have a little north of 200 patients enrolled as of the end of March. That number is now closer to 213. Just because this continues to accrue over time. And about 140 of those patients were actually through sequencing. So patients, can be nominated but not accepted onto the protocol or approved, as I mentioned, and you can see the relative numbers here in the graph, and there are a variety of reasons for that. And then the other question that you may be asking yourself is, well, this all sounds really great. You're doing a lot. What does it all mean in terms of medically meaningful information? So actually I have a breakdown here for the 188 patients that have been evaluated at this point in time. This is fairly recent data, and essentially shows that the reasons that I mentioned earlier in the light blue boxes for why patients may not be consented, that would not be a surprise to anyone, and other reasons why patients, even if consented, may not be sequenced. And again, these are not surprising reasons. Everybody who does this sort of has this kind of a drop off. But the most important thing I think to focus on here is at the bottom, these yellow boxes basically give you a breakdown of when we are finding what we call medically meaningful findings. So from this combined RNA DNA analysis that I just walked you through. So are we getting a diagnostic information? Are we getting prognostic information? Or are we identifying therapeutically relevant results that can be, again, confirmed clinically and then written into the patient's medical record for use by the oncologist? And so numerically, you can see the breakdown here across germline predisposition, fusions, which we talked about, targetable expression, which we talked about, copy number, as well as chromosome level changes in copy number, such as whole chromosome or arm level amplifications. And then finally, medically meaningful somatic variations, so point mutations that may be illustrative of the likelihood to respond to targeted therapies, or even a high tumor mutational burden that might be indicative of the likelihood to respond to checkpoint blockade. If you sum these all up as a percentage, we currently estimate that about 93% of the patients that we're testing, or have tested to date rather, are receiving one or more medically meaningful results that can be important for their care. So I think that's probably much higher than I would have expected it to be. And we can talk about the nuances of that during Q&A if you're interested. What I want to do now, just for the remainder of the talk, is to walk you through a couple of vignettes to really illustrate this protocol in action and sort of talk about some of our novel findings and therapeutic interventions that have been achieved for many patients, but I'll just focus on a couple. This first patient's story is a child at 1 year of age, originally diagnosed in 2013 with a meningeal sarcoma. There was some suspicion of syndromic or susceptibility because of his father's medical history, dying of early onset colorectal cancer at age 39. And the patient in particular had received a gross total resection in 2013, followed by proton therapy, which would be indicated by such a young child. We began this study of his tumor in 2018 when he was diagnosed with a recurrence of this meningeal sarcoma. So let's walk you through those data as we found them. Here's just sort of a MRI-based view of the tumor at 2013, the gross total resection that was achieved. And at this time, in 2014, I was not at Nationwide Children's Hospital, and the Institute for Genomic Medicine was not a thing. And so because of this rare tumor type and the child's age, the DNA was sent out to a commercial testing company. And their results essentially were a pathogenic mutation identified in this intestinal cell kinase gene and a VUS in platelet-derived growth factor beta. So again, as I mentioned, the patient received proton therapy and had a recurrence in 2018 following that therapeutic interval. The gross total resection from 2018 was studied on the IgM cancer protocol that I just told you about. Here's what we found. So I mentioned these results from a prior testing. It turns out that the testing that was done was not only done on the tumor material, as is fairly common. And indeed, when you test both the germline and the somatic, this intestinal cell kinase shown on the left in an IGV view is in fact a large deletion that is present in the germline and the tumor. This is essentially not thought to be important in the underlying predisposition to cancer, by the way, but just shows that the value of sequencing the normal as well as the tumor. And the really important result is on the right-hand side, which is that the VUS that had been reported in PDGFR beta was actually an in-frame deletion. And if you look at the graphic on the right-hand side, that shows you the structure of PDGFR beta. And this occurs lower down in the protein in this area called the juxtamembrane region, which is actually frequently found mutated in cancers that have PDGFR beta mutations. And it essentially is turning this RTK, this membrane-bound receptor tyrosine kinase on permanently. So this is the signaling mechanism that's driving this child's tumor, in other words. If we look at the level of RNA expression in this patient, compared using the approaches that I told you about earlier to all of the other patients on our panel, on our cohort rather at the time, you can see the red bar refers to this patient's expression of PDGFR beta, and just in the blue bars, a couple of other patients previously described with this same type of juxtamembrane alteration, although maybe not the exact same variant, but definitely showing that overexpression of PDGFR beta is the result of that type of alteration. We provided this information back after we produced a Sanger-based clear confirmation and to the provider at Nationwide Children's Hospital, and worked with him to identify the fact that likely treatment with dasatinib was going to be appropriate in this recurrent cancer for which there is not a second standard of care beyond what had already been utilized. And so the patient was able to receive dasatinib therapy. And my MRIs are a little out of date here. We do have a February 2020 MRI from this patient that is also showing no evidence of disease. So this is now an 8 going on 9-year-old boy who has really responded very, very well to the indicated targeted therapy for his meningeal sarcoma. I want to take a little bit time here now to talk about a second case, which is really one of these longitudinal cases that I talked about. So multiple samples involved. And a bit of a diagnostic dilemma. This patient came to our attention from our neuropathologist, Dan Bouet, who had performed a diagnosis at this patient's original cancer discovery in 2012, labeling this medulloblastoma. The patient recurred in 2015. Again, the pathologist, Dan Bouet, had evaluated the samples from the secondary cancer and called this a recurrence of medulloblastoma. But in 2018, this patient recurred, and in fact, Dan was not 100% sure that this third occurrence was a medulloblastoma. So he brought this patient to our attention and said, I think it would really help my diagnosis if you could study this patient through your IgM cancer protocol. So we actually had access to the 2012 tumor and of course to the 2018 resection. I'll walk you through now a series of results using the tumor normal DNA here, showing copy number variation. On the top panel, all of the blue data essentially are the copy number probes that I told you about earlier, reporting out for each of the chromosomes, and they're numbered across the bottom. So this is the entire genome being represented in terms of copy number diagram. You can see the 17 P-arm deletion, for example, which is characteristic of medulloblastoma for this subtype, which is group 4 beta, as well as a chromosome 4 amplification, neither of which are available or seen rather in the recurrent cancer from 2018, which is the lower panel of copy number data. Here's a comparison of just the somatic mutations that were identified. The recurrent tumor here is on the x-axis. The primary tumor mutations are represented according to their variant allele fraction on the y-axis rather, and you can see that there's essentially no sharing really whatsoever of the variants between the two tumors, which would indicate that they're very, very different, i.e. not the recurrent not derived from the primary. The only sort of interesting finding in the recurrent tumor from 2018 is this PTPN11 variant that's indicated that again is unique to this recurrent tumor. So what do we know about this sample or this PTPN11 variant? This is a fairly well-known a pathogenic gain of function variant by a variety of sources. They are shown here, but this has not really been found per se in medulloblastoma, primarily in hematologic malignancies, as you can see the data from COSMIC, but seen in a multitude of different tumor types writ large. And so this actually turns out to be very interesting for reasons that I'll show you in just a minute. One of the comparisons that we also did was at this level of outlier expression, which I talked about earlier. You can see here on the left-hand side, outlier expression for the primary tumor has a number of highly expressed genes that are diagnostic of medulloblastomas. None of those are present on the right-hand side. And indeed, this PDGFR alpha overexpression turns out to be important in the context of understanding the PTPN11 variant that we just saw a minute ago. So PTPN11 goes by another name, which is SHP2. You can see on the diagram on the right-hand side of the slide that it signals just downstream of PDGFR alpha. And so this is really a driver interaction that's going on in this patient. And for the purposes of druggability would actually be important because there are drugs that are available to shut this down. But let's dive down a little bit more into the initial question, which was what type of a tumor is this? What I'm showing here now is this treehouse-based principal components clustering that I talked about. This is one of the tools that's available at the Treehouse data site. And what you can see quite clearly from these black arrows indicate that indeed the primary is a medulloblastoma. It's grouping down along that group of blue medulloblastomas, sort of in the center lower part of this PCA diagram. But the recurrent cancer is grouping up above, maybe more closely with a diffuse group of gliomas, generally described as gliomas. So different subtypes and different glioma types, but it's definitely not a medulloblastoma based on the RNA expression profile. And again, you can see just an example here. This is now a heat map instead of a principal component. But what we're doing is we're taking 101 medulloblastoma patients from that Treehouse database, subgrouping them in comparison to the primary and recurrent cancer from this child, and just for the purposes of comparison, adding in another patient that we had studied previously with a high-grade glioma. What you can see quite clearly is over on the far right-hand side of this heat map, both the high-grade glioma comparator and the recurrent cancer from this patient, but not the primary, are grouping very closely together. And so that indication really tells us that these are more falling in with sonic hedgehog type medulloblastomas, whereas, as I mentioned earlier, this patient's original diagnosis was in group 4 beta. And we know from years of medulloblastoma recurrence and subtyping that the recurrence is typically in the same subtype or subgroup. So based on all the information that I just told you about, we were able to go back to Dr. Bouet and basically say, look, these things all tell us that this is not a medulloblastoma. We went through this data with him carefully, and he ended up agreeing with us and did change the patient's medical record to reflect the fact that this was not a recurrent medulloblastoma, but is instead a second malignancy. And we also used the CyberSort comparison that I told you about earlier, just to compare these two in the context of what has changed about the immune microenvironment. Again, telling us maybe that there are differences between the two above and beyond the DNA and RNA-based differences, again, what you can see here is the lower panel, and with the corresponding colorful key on the right-hand side, reflects the immune components of the 2012 medulloblastoma, which have exploded essentially in the 2018 glioma, to include significantly more CD8 T cells, as well as in the very large sort of purple bar M2 macrophages. And so macrophage infiltration is something that we are now routinely seeing in these recurrent brain cancers across a very large number of samples, which I don't have time to talk about today, but this just falls into line with much of what we've seen before. The bottom line is that, according to Dan's analysis, combined with our evidence as well, this child has a second malignancy. It's likely related to the fact that in the earlier occurrences of the medulloblastoma, radiation therapy was utilized to treat the patient. And this is just showing data from a study published in Neuro-Oncology a few years back that shows us that about within 10 years of this original diagnosis and radiology treatment of medulloblastomas, that about 4% of patients recur with a radiation-induced secondary tumor. And so this, again, I think is a motivation for us to try and begin to explore areas that do not involve cytotoxic chemotherapy or radiation therapy, especially in ways where we may be able to apply a targeted therapeutic more early in the disease course and avoid some of these negative downstream impacts of treatment that we see. I just want to finish this by saying, I mentioned JL33, this high-grade glioma. We have also studied this patient's primary and recurrent CN copy number, as well as mutations and RNA writ large. And this is just a comparison to show you that in both of these cases, we had very different mutational profiles shown on the left-hand side of the slide, very different copy number profiles shown on the right-hand side of the slide. And so this can be important information just in terms of the relationship between the primary and the recurrent cancer, where in both cases, these were very different tumors. And then this is just a second patient data from a second patient with essentially the same story, a primary medulloblastoma, a radiation-induced secondary glioma, and where they both are grouping in the treehouse comparison that I walked you through earlier, the PCA plot, again, both grouping in the primary setting with medullos and in the secondary malignancy with gliomas. So we've now seen 2 examples of this in our experience so far. This is just a slide to show you that, hey, we're there every Friday morning at 7:30 working with a neuro-oncologist. We also present at the sarcoma tumor board as well as the generalized hem-onc BMT tumor board. But in this particular setting, we actually have the screen on the right, which is showing the data, much like I just showed you from a PCA plot, where the radiologist is looking at the child's historical and most recent scans. And so this has become a really dynamic environment where the genomic information that's being produced is very welcomed by our providers, and we have some great discussions around it. And this just really shows you that, building on what we're doing now, the cancer analysis is just part and parcel of one of the many ways that genomics can play a role in the context of the treatment of pediatric diseases. And you can sort of read these different blue boxes, which map out our strategic plan over time to roll out genomic analysis for the improvement of diagnosis, and in some cases just really understanding exactly what went wrong. What is the underlying genetic causation for the child that we're looking at today? If you look at the bottom row, you'll see a box labeled epilepsy, which includes enhanced whole exome analysis and RNA-seq. And I just have like 3 slides to show you how we're taking that tumor normal paradigm that I just talked to you about for the last 30 minutes or so, and expanding this out now into profiling of brain tissue that's essentially been resected from patients with intractable epilepsies. So some 30% of kids with an epilepsy diagnosis go through multiple treatments and don't respond with a diminution of their epilepsy seizures. And in some cases where this is really debilitating, they can go through a process that is essentially electrophysiological mapping, shown on the right-hand side of this slide, that's performed during sleep when the brain is least active, to identify the areas of the brain that are most affected by and are the source of these seizures. If these are in non-critical parts of the brain, they can actually be removed in a resection process during neurosurgery with curative intent. And so we've really sort of latched onto this as a means of applying the genomics to help understand and to help our parents as well as the epileptologists really understand what's happening in these patients. And so I'm really excited because we just got our first case published. It's this child, a 3-year-old male with treatment intractable epilepsy. Blood-based testing was negative from the Ambry genetics panel. And so he went through this process that I just described. And is sort of exemplified here, where the electrocorticography mapping was performed, subsequent neurosurgery, and then the resected parts of the brain were evaluated by pathology, analyzed accordingly, and then handed to us for DNA and RNA extraction and this exome versus RNA-seq that I told you about. I'll just cut to the chase. This is what's was found and will be published soon in Neurology Genetics. And essentially what we were able to show is that this patient has a mutation, a frameshift mutation that is a somatic mosaic in this gene SLC35A2, which is a known epilepsy predisposing gene if inherited through the germline. This is not the case for this child. Clearly, during some point in brain development, this mutation crept in. As you can see from the red arrows and the percentage signs associated with them, in different regions of the brain, this mutation is present at different levels, essentially as a somatic mosaic. So it's in some cells, but not all. And indeed, when you look at the percentages and overlay the pathology evaluation of these tissues and overlay on top of that the electrocorticography measurements, what we find is an exact corroboration between the percentage of the mutation present in the cells and the severity of the epilepsy seizure as obtained through the electrocorticography, as well as the derangement of the neurons as noted by our pathologist, Chris Pearson, who evaluated this patient's samples. So if you think this is a one-off, we thought so too for a while, but we've actually now studied 36 patients, 30 of which are shown here, using this protocol. This is again an IRB approved protocol, of course, to study these patients' tissues. And you can see that in the majority of these patients, we've got no findings, but in some 34%, we have a validated somatic finding. In a few cases, we actually were able to identify just a germline VUS, but that would have been identified through a panel anyway. So this has turned out to be a pretty interesting study. We have 2 new patients that we're just in the process of writing up and trying to publish as well from this study. So I'll finish by just saying that I think these clinical genomic assays can add some important clinical evidence, and they can be important for treatment planning, diagnosis, and prognosis, as I've tried to illustrate. I really feel like the combination of DNA and RNA is so powerful. I tried to illustrate that in the remarks in terms of what we're finding, and admit that the RNA data still remains a bit of a confounder, but we will continue to pursue it. And really, I want to try to emphasize in particular, now that we have funding for a childhood cancer database initiative through the NCI, that's getting ready to announce awards soon, from what I understand, that we've got to continue to share data because of the rarity of pediatric cancers, because of these vignettes that I've told you about. I think the more we can share that information, the better off we're all going to be in terms of being able to offer our pediatric patients the best care possible. So I'll just finish by thanking everyone, and it does take a lot of people, as you can see from my very full slide. In particular, just want to acknowledge the enthusiasm of our NCH collaborators, shown on the right-hand side of the slide, across the neuro-oncology and epileptology areas, and especially want to thank patients and families because of their bravery for volunteering for these studies. We're able to do what we're able to do, and so that takes a lot of courage. And then lastly, I want to thank the Mark Foundation, based in New York City, for supporting my postdoc, Katie Miller, shown there in the middle of the slide under the computational analysis. And that's it. And I'm happy to try and address any questions that came up during the talk.
Well, Elaine, thank you. That was phenomenal. And I think it really
Thanks.
no, it was great because it really eloquently underscores how translational genomics really is, and how it can not just impact our understanding of disease, but how we actually treat it. So it really resonated. Thank you. As we're waiting for people to sort of enter their questions, I thought I'd take my prerogative and ask you just a couple to start. First, your tissue source, FFPE or fresh frozen?
Yeah. In an ideal world, everything would be fresh frozen, but we really do evaluate formalin fixed material because often that's the only thing that we can come by. We are tuning up this pipeline to try to subtract out some of the artifacts that come along with the FFPE as we move it on that transition that I talked about into the clinical setting. So we'll continue to accept both.
And is your source for germline adjacent normal, or do you have blood or spit? What do you use for that?
We typically get blood. We have encountered, as I'm sure you can relate, Ken, a number of patients that we can't get blood from for a variety of reasons. And so in those cases, we will typically use a buccal swab, if that's a possibility, or spit at a minimum. It's not as, it's interesting. Sometimes the quality isn't quite as good. We have encountered that. But actually, by doing the exome capture, you eliminate one of the confounders for using buccal material or spit, which is the large amount of microbes that come along for the ride. Which, if you were doing whole genome sequencing, you would be sequencing all of those. So that's actually okay, but we do sometimes encounter some problems with quality coming out of those types of samples. I also mentioned, or maybe didn't mention, but it was in the diagram, we do consent the parents if they are willing to consent. We don't actually study them by sequencing only in the case where we're identifying a germline variant. Would we then go back, and that's typically a buccal swab from those parents. We would then go back and spot check for inheritance patterns from a germline finding.
So if you identify a BRCA mutation in a kid, report it?
So that's one of the nuances of the consent form. Carrier status is optional, and parents can either agree to learn it or not, for themselves as well as for the child. Now, I sort of focused on kids because that's much of what we see at our hospital, but as you can well appreciate, we do have a considerable number of AYA patients. So there, obviously they can consent for themselves and agree to carrier status, et cetera. But all of that language is definitely written into the consent. And there are specific places where people either sign or say, no, I'm not going to agree to that, depending upon how they feel about it. So again, the importance of a genetic counselor to really walk parents and patients through those thorny questions.
And then my final question before I turn to the questions in the group is relates to exactly that, the AYA group. We know AYA exists. We know that AYA adults do better on pediatric protocols. You might expect that implies that there's perhaps a significant subset of adult patients with pediatric signatures. And I'm wondering if you started to take advantage of the other databases to perhaps identify those, in both the known AYA cancers and perhaps unknown ones, and maybe sort of think about putting them in pediatric trials.
Yeah, so we have we reach out to a lot of databases. I really just illustrated PCAN because it's so pediatric centric. We have many other, C-bioportal in particular, depicted in our search space. Um, and it is really interesting. I think we don't understand enough about AYA cancers from the standpoint of large-scale discovery, I would argue. They're at the fringes of any pediatric or any adult setting type cancer discovery project, but the preponderance of adults that have been studied with cancer are relatively old. The preponderance of kids that have been studied are relatively young, and I just feel like there's a huge gap there. This is fresh in my mind because we just studied a 20-year-old patient with medulloblastoma, which is exceedingly rare. And so sometimes we're just wandering in the dark. At the end of the day, yeah, if we can find something, it's fairly straightforward to get that patient onto a study. We actually just studied a young man with very advanced, I think, second or third recurrence of I can't remember the exact diagnosis, but a CNS cancer who has an FGFR fusion that essentially qualified him to get onto a trial of the Janssen drug. So we went back to an old specimen. We were able to confirm that fusion, and he's now enrolled onto that study. Erdafitinib, I think, is the name of the drug. So we can do that. I think he's, again, 20 or 22 years old. So we'd definitely try.
Okay, so now I'll turn to the questions from our audience. The first is from Irene. It's regarding the patient with the two primary tumors. Radiation therapy can obviously be one cause for the second tumor, as you mentioned. However, is the occurrence of two primary tumors in early life still raising the suspicion of germline predisposition, for example, of a gremlin mutation impairing DNA repair?
So just to be clear, these were not coincident tumors. This was a series. So the primary was indeed a medulloblastoma, had all the hallmarks of a medulloblastoma, was treated accordingly. This 2018 tumor was not sort of growing at a different, at a different site more slowly. For example, I suppose you could argue maybe it was. I guess the fact that we've seen two of these patients, the fact that it's been documented by a COG study that radiation does induce the second tumor, would argue against it. But from a formal standpoint, because they were so genetically different, I couldn't argue against that successfully. I think the only thing that I think the mutation that you're talking about, though, was not in the germline, because everything that I showed was somatic. So we would have indeed studied this patient's blood, and if there had been a germline, mutation to report, I would have shown that. So we didn't see that. In the first patient that I talked about, there was definitely a suspicion of germline predisposition, but that was never substantiated, again, by anything that we were able to find from the patient's blood. And even though the father was deceased, interestingly, there had been a preserved specimen of his that we could test for germline susceptibility, but we were never able to get that. So
there you have it.
Yeah.
So our next question is from Anne Bocock. Hi, Elaine.
Hi.
For your pediatric cancer pathogenic mutations, what proportion are sporadic versus germline mutations? And have you also looked for germline mosaicism in their parents? Also, for heritable mutations, how often did parents develop other cancer types?
So I think most of these are sporadic. We don't understand why, certainly. We have not evaluated for like gonadal mosaicism, which does happen, obviously, because we don't have the right sample, and we're not sequencing, again, sort of to that level of depth. We do generate a high depth focal assay of the mom and dad's germline if they're made available, and if they consent to testing. We could technically find a somatic mosaic in that. It hasn't happened yet. So those are good thoughts. We're looking for them, but we just haven't come across them yet. What was the second half of the question, Ken?
The second question was, so germline mosaicism, then also for heritable mutations, how often did parents develop other cancer types?
Oh, right. So, to quite frankly, like the first patient with the meningeal sarcoma, some of these patients come to us with a family tree essentially put together by the genetic counselor and with some significantly known or suspect, I should say, germline predisposition. Um, we find that most of the time, but not always. And as I'm sure everyone can relate, that remains a vexing problem that I think nobody really understands. But usually we're successful in coming up with it. I would say probably 75 to 80% of the time where there's been something suspect, we've been able to uncover it, but not 100% for sure.
Right. I mean, peds, don't forget, everybody's young,
Yeah.
so they haven't had time to develop things. Then there's anticipation, and then there's variable, differences in penetrance.
Well, I think too, to your point, Ken, as well, we still don't understand the totality of genetic susceptibility. I mean, there's a very interesting paper recently in Nature in the pediatric setting, small subset of medulloblastomas, but the suspect gene and mutation were really in a transcription factor, cofactor, right? But when you work your way down the mechanistic pathway of what that entails in terms of cancer susceptibility, like it makes sense. But these are sort of the little bits and pieces that we chip away at to try and learn what new kinds of mutations actually can lend themselves ultimately to the cancer susceptibility. So I think there's a lot more sleuthing to be done.
Our next question is from Marty Walsh. Great talk and enjoyed. Can long read approaches, i.e. PacBio or Nanopore, alleviate and complement the challenges with mapping the fusions and amplifications losses, which could remain as confounders for some sampling for both RNA and whole exome, whole genome? Or do you believe machine learning pipelines will get to the point of high resolution mapping from Illumina after having enough attempts to train?
I think it's the former, and we have, it's just not enough time, but we have great evidence of both very complex rearrangements being solved at the DNA and RNA level in a few of our cancer patients that have gone through the long read sequencing. And, in particular on the Pacific Biosciences instrument, we're now in the realm of being able to make a genome library out of about 3 micrograms of DNA, still a relatively large amount, let's say, but doable from a large gross total resection of tissue. And RNA through the IsoSeq protocol is working very, very well. So I have a nice story that I think I presented for one of the ACMG talks that I gave around solving this very complex rearrangement in one of our cancer patients. I think it was in PDGFR beta actually, that we were able to show what resulted was very similar to that juxtamembrane indel for the patient that I talked about, but in this case was just a very complex rearrangement around exons 11, 12 of the same gene. So I think as this price point falls and as the informatics tools are more facile for the long read technologies, that's really going to become an important part. And maybe even in reference to the earlier question about, if there's suspicion of germline susceptibility and you don't find it, maybe that's why. So it's a really interesting space, I think, right now.
The next question is just to confirm that you said that in pediatric brain tumors, fusions are more relevant than point mutations?
Oh, I don't think I said that. I just said that in general, when we're looking for fusions versus point mutations writ large in pediatric cancer, fusion drivers are significantly prevalent. I would say, even more so than in adult cancers. Like you could pick out some adult cancers with known fusion drivers, but the relative, numbers are much higher in pediatric cancers. Certainly in ALL, as we know, Ken, but in all types of cancers, EWS fly, in Ewing's, Kia-BRAF fusions and pilocytic astrocytomas. I could go on, but I think writ large, they're much more frequent.
Our next question is from Emily Bernstein. Are ATRX mutations common in medulla, as they're found at high frequency in other pediatric cancers such as neuroblastoma and osteosarcoma? Are these missense? If so, I guess, are these missense or gene fusions? And then in parentheses, I noted ATRX on one of the slides. Thank you.
You're probably paying more attention than I was. No, I'm just kidding. ATRX, I'll be honest with you, we haven't seen a huge number of advanced neuroblastomas in our practice so far. I think you could count it on sort of two hands. So I don't know that I could really speak intelligently about prevalence, fusions versus, one versus the other, just because we haven't seen that many. Not sure why. I guess they're either going other places or they're being addressed adequately by the existing therapies.
Next is thank you for a beautiful seminar. And that's from Kiprin01. And then finally, there's a question from Ben Hopkins. Hi, thank you so much for sharing your program pipeline with us. When you say that 186 had medically relevant, I think that was the term in the slide, findings, can you unpack what that means? And how many of these patients got a different therapy as a result of the pipeline?
Yeah, so great question. I'm sorry if it wasn't clear. What that total number in aggregate means was broken down in the boxes along the bottom of the slide. So medically meaningful as we define it is that it refines the diagnosis or makes it definitive, that it provides prognostic information, which of course changes treatment, because if we know that a specific tumor is classified as more or less aggressive, that's important information to the oncologist in terms of what the aggressiveness of the treatment is dialed up or down accordingly. And then lastly is just sort of any therapeutic relevance that can be obtained from either the fusions, the point mutations, copy number, et cetera. So that's really the way that we define medically meaningful for that 186 set of results. There is some interplay because some patients provide more than one piece of medically meaningful information. So, but if you just look patient by patient, as I mentioned, we do feel like about north of 90% of the patients actually get at least one type of result that's prognostic, diagnostic, or indicates therapy. The second half of your question is also really interesting because, of course, all of the things that I stated in the first slide are reasons why a provider could look at a series of pieces of evidence, much like I talked about today, and basically say, well, gee, I just don't know if I want to do that because, and all of the